So with Aldevron, we've been around for about 21 years, co-founded by Michael Chambers and John Ballantyne in sunny Fargo, North Dakota. So we'd love to have people come out and visit us. It's really warm there now, so I think that's the time to do it. So today we're just going to talk a little bit about standardized plasmids and large-scale production for gene therapy. A little bit, never know quite the background of the audience, go through pretty quickly how plasmids are used in gene therapy, talk about these plasmids that are common to both AAV and lentiviral productions, some of the advantages of the standardized plasmids we'll talk about, and then scalability of the plasmid production in our facilities. So in gene therapy, there's multiple ways that plasmid DNA is used. So direct injection is one, usually complex. We support all of these different areas. Direct transfection, where cells are removed from the body and plasmid is introduced. In vivo raw material, so a viral vector is administered in vivo. It's raw material for that vector, or ex vivo raw material when the vector is used on cells outside the body. Also used as a template for mRNA production, where the mRNA is the API, and also where our mRNA is transfected to a cell outside the body. So a lot of different uses for plasmid that we support. Today we're going to focus on these two areas where it's a raw material for a viral vector. So in the case of recombinant AAV, probably one of the most common viral vectors. Just a simplified view of the production process. On the top, we start with a bacterial cell bank from which we make plasmid DNA. And then there's a mammalian cell bank, probably most common is HEC293, that are combined together, and the plasmid is transfected into the mammalian cells, and then that vector is delivered directly to the patient, carrying the gene of interest to treat the disease. So another sort of example, probably multiple people have stolen this off the internet. You have your gene encapsulated AAV, and then the point is for that AAV to deliver the gene of interest to a target cell and then express either on the surface of the cell or secreted protein to treat some disease within the patient, either a gene replacement or other kind of treatment. So the most common method of manufacturing AAV is triple transfection, where you have the transgene plasmid, also sometimes referred to as cis plasmid, with your gene of interest, and a rep cap plasmid, and then the add helper plasmid. And these are called packaging or helper plasmids, with the expression plasmid co-transfected to get your AAV vector. So this helper plasmid is common to all RAV production. So regardless of the serotype or transgene, you need these helper functions, which are adenoviral genes, in order to produce the AAV. And this is the plasmid that we've worked with colleagues at Asclepios Bio to develop, PLDX80. So this is a plasmid that we now have available at research grade and at clinical grade as an off-the-shelf product for our clients. So the plasmid that we manufactured, expression data for RAV, recombinant AAV luciferase. So the PLDX80 shown as either produced with mini preps or an industrial process showing equivalent expression to other helper plasmids available on the market. So this plasmid has some really significant benefits for those of you who are struggling with being able to produce more AAVs, being able to make AAV at scale. So this is based on the PXX680 that Jude Samolsky published in 1998 with Zhao Zhao. We worked with Asclepios Bio to get access to this, and Jude really sees it as an opportunity to contribute to the field. It's royalty-free, including commercial products. So one of the areas that can be a challenge is IP with AAV. So this product, when you purchase it from Aldevron, there are no subsequent milestones, royalties, or anything like that. It can replace any helper plasmid. The field is moving on to kanamycin resistance over ampicillin. This has kanamycin resistance already. It's immediately available. We offer free samples for research applications, and you get a consistent, reliable supply of this plasmid because we make it at scale. So moving over to lentivirus, similar process here. We have bacterial cell banks with plasmids and mammalian cell banks making the lentiviral vector. But then that is typically administered to patient cells outside the body, although there are certainly programs where lentiviral vectors are administered directly. And then the cells are infused back into the patient. Again, a more detailed view of this where we have the virus made in the laboratory transducing these cells outside the patient and then re-administering them. So again, a transfection process here. We've got the transgene plasmid carrying the gene of interest, and then there's three called packaging plasmid, VSVG, GAG-PAL, and REV, that are available, that need to be available, and these four plasmids are transfected into the lentiviral vector. So in this case, we have three plasmids that are common to all recombinant lentiviral production, the VSV, GAG-PAL, and the REV. So these are plasmids that we, again, have available as off-the-shelf products, both at research grade and clinical grade. So they're available to ship immediately. And I haven't gone into a lot of detail, but we also have the expression plasmid into which that we can clone your gene of interest to give you the complete set. Of course, that's going to be a custom manufacture, right? So we're going to manufacture that on a per patient or per program basis. But the other three plasmids are available off the shelf. Again, that consistent supply. So we partnered with Oxford Genetics for these plasmids, and this shows their data. Showing the Ph.D. lenti system compared to other company plasmids, both in adherent and suspension 293 cells. So suspension 293 is definitely becoming available and in use quite a bit for viral vector production, showing that we have as good or better performance than other plasmids that are available. So these are key plasmids for lentiviral production. They've been codon optimized and with minimization of homology and intercassette homology. Again, they're royalty free. You don't have to worry about the IP issues around these plasmids. Replaces any third generation lentiviral plasmid system. So even though for the other systems that you've started out with, we can use these plasmids. These again have kanamycin resistance, immediately available. And again, as we discussed, the expression backbone is also available. So if you're going to standardize something, you have to have standard specs. So this is something that the field kind of has to adjust to if we want to get to the point where we can really industrialize this manufacturing process so that we can make it reproducible, ensure reliable supply chain, and meet all of those requirements. So the specifications highlighted in blue are really the important ones which drive cost and time. So we've chosen a set of specifications that we believe meet the needs and requirements of our clients, yet optimize the production efficiency so that we can produce these at scale and drive down costs as much as humanly possible. We're always looking to the industry to get more advice on this, and certainly continually evaluate these specifications and are open to different specifications. But even for custom manufacturing, we really ask clients to think about these specifications and what is necessary for their programs, and that these specs aren't just some legacy handed down from prior experiments, and that there's really data that suggests that these are the required specifications. And these are both for the lentiviral and helper plasmids for AAV. So just one quick slide on scalability. So last year, Aldevron opened a 70,000 square foot facility in Fargo, North Dakota. In that facility, we have two production trains. One has a 300-liter fermenter. It's gone quite well. We've had multiple runs with our 300-liter fermenter that have been successful. We've used this to manufacture the ALDX80, and we're about to spin it up to do the lentiviral plasmids. And we show scalability from our 30-liter reactor here, or fermenter here. So we're confident that we can produce these plasmids consistently and make them available on demand for our clients. And I'll give you a little spoiler alert for the people in the room tomorrow. We're going to announce expansion to 1,000-liter fermenter capacity. So again, scaling up to really make these things available to our clients as needed. So we have a poster with this data as well that'll be Wednesday evening, 5 to 6, in the Columbia Hall right out here. You can also download it. I've got a very long URL here, but if you just go to product overviews on our website, you can download this poster. We also had a press release earlier this week, and you can download the poster from that press release as well. So thank you very much, and we look forward to meeting your needs with standardized plasmids.